and it's a delight to be here, especially to escape on the cast of media and all that influence. So let's see some solace in something. So I have been, and I had a tough time trying to figure out how to not bore people who know some of my stuff and yet make this accessible to people who don't, and so I really bad. Okay, I just move it closer. Quiet, wave your hands. Okay, so I have for a shocking long time been making the following set of claims. That the human brain, and by brain I usually mean cortex, I'm a very cortical chauvinist, but the cortex has many regions with distinctive, often very specific functions. And that includes regions that are selectively engaged in perceptual processing of faces, places, and bodies. So, for example, if you use the face area right here on the bottom of the brain, here's an example of some functional MRI data showing that it responds much more to faces, that's the blue bar, than to any other class of stimuli. Those are the various other colors. So, and same deal for the place selected and body selected regions and lots of other cortical regions. But it's not just high-level categories of visual information that have selective responses in the brain. Even regions that are specific for abstract, kind of quintessentially human functions, like understanding language. So, for example, these red regions, this is very schematic, but in the left temporal and frontal lobe, regions that have long been implicated in language processing, actually for hundreds of years, can now be studied with great precision with functional MRI. And so Ev Fedorenko has developed ways to identify those regions in each subject individually and then measure their response to an arbitrary number of different tasks. I think she's run like 100 different tasks by now. And what she sees in experiment after experiment is that although the regions respond strongly when you read or listen to sentences, that's the black bar, not when you read or listen to non-word strings, showing their selectivity for language processing or their engagement in language processing, they respond basically not at all to a broad suite of other tasks, from mental arithmetic to logic problems to listening to music to thinking about what other people are thinking. Those activities produce essentially no activity in those regions, showing that it's not just another selective region of the brain, but also showing that language is not the same thing as thought in the brain, something that I think is kind of a cool discovery. Another distinctively human function is music. And so we've found, schematically shown with these little orange blobs here, first with functional MRI on the top, scanning people listening to 165, I think, different natural sound clips. All the blue and purple bars are the responses to different kinds of music, and all the other colors are responses to non-music sounds like dogs barking and toilets flushing and people talking, etc. And so that shows remarkable selectivity of the response to music sounds, even though music itself is an astonishingly heterogeneous category that spans from, a classical flute solo to a heavy metal band to an opera to a drum roll, right? Any of those things strongly activate that region. And this has been validated with intracranial recordings. Here's the response of a single electrode to the same stimuli in a patient undergoing neurosurgery. And to me, the most astonishing of these selective regions is the work of Rebecca Sachs, showing that this region right here in the temporoparietal junction, like right there, more in the right hemisphere, is extremely selectively engaged in thinking about what other people are thinking. It's not enough to think about another person's thirst or hunger or pain. It's not all mental states. It's only when you're thinking about the contents of their thoughts or their percepts that region is engaged. And if that seems at first completely absurd, reflect on the fact that this is kind of the essence of being a human being, is to allocate a lot of your mental effort thinking about what other people are thinking. And so perhaps it's not shocking that we have specialized machinery for that. Okay, so that's just a whirlwind overview of some of my favorite selective regions of the brain. I want to hasten to say that I do not make the claim that all parts of the cortex do something ridiculously specific like this. In fact, we know that these white regions here, big swaths of frontal and parietal cortex,
are very generically engaged in almost any difficult task at all. You just do something difficult, they turn on, they don't care what it is. So there's some cortical regions that are very specific and others that are not. I further want to point out, these are things that my colleagues kind of hassle me about. Kanwisher, you think everything's specific? No, I don't. Similarly, these regions don't act alone. Of course they don't act alone. No part of the brain could act alone. Each of these regions is connected to and presumably interacting with lots of other brain regions. And all I mean by pointing out these selectivities is that little patch has a specific function, however it does it and whoever it interacts with as it does it. And so why do I care about this? I'm a psychologist, not a neuroscientist at heart, and I care about this because I see this as a very rough initial sketch of some of the basic pieces of the human mind. And I think that's pretty cool. But I also think that this picture really is very, very rudimentary, and really what it is an invitation to ask all the next questions, of which there are many, and of which I will grapple with just a few today. So first, I'll dig in a little more on what I mean by the specificity of these regions, and consider the question of whether their specificity is not just in their response in functional MRI, but also their causal role in behavior. I will consider the question of what other mental functions might have specialized machinery in the brain, the question of how does all this organization arise in development, and the question of why is that a good way to design a brain in the first place? And I won't answer those questions. I'll kind of dance around them and offer humble tidbits of data that might eventually constrain answers. So let's start with this first one. To my surprise, this question is still considered controversial, whether these regions are really specific. So to give you a few examples, here's a quote from Lisa Feldman-Barrett a couple years ago. She says, most brain imaging studies make 3 questionable assumptions. Mental events are localizable, map uniquely to dedicated brain circuitry, and are independent of larger context. These 19th century views need an update. Okay, that's Feldman-Barrett. Here's Luis Pessoa. Neuroscience tends to study parts of the brain separately. This standard decomposition adopted by neuroscientists requires an organization that's fairly modular, which is inconsistent with the principles of anatomical and functional neuroarchitecture. I find these quotes puzzling, because actually there's a lot of evidence for functional specificity of many regions of the cortex. So I'm going to take a little time to show you 2 of my favorite studies that argue for the selectivity of a couple regions to give the flavor of the kind of evidence, kind of reason we make these claims. It's not just we make the claims because we think it's fun. There's decades of research behind it. Okay, so the first one makes use of artificial neural networks. Much to my surprise, I find myself running a lab full of people training artificial neural networks. I am decidedly not a computer scientist, but turns out these things are really powerful. So in case you've been living in a cave for the last 15 years, this is a very standard, simple, convolutional neural network, an artificial neural network that's just composed of a bunch of units arranged in a set of layers. This one trained to recognize objects. This is AlexNet from 2012, the one that really broke on the scene and changed the whole field. So you stick in an image and it creates a cascade of activations through layers of the network, ultimately resulting in a choice of one of a few hundred or 1,000 different possible object categories, okay? And of course you train it up with lots of images, and there's many ways to train it, and we're skipping all that for now. So you take a trained artificial neural network, and so I'm going to lead to how we can use this to ask questions about specificity in the brain. But so how do you do that? This is based on prior work from my colleague Jim DiCarlo, working with Dan Yammans, in a really fabulous paper from 2014. So they wanted to use one of these trained artificial neural networks as a predictive model of what a single neuron in monkey visual cortex was doing. So they record from a neuron in monkey visual cortex and its response to a few hundred images. And now they want to say, can we make a predictive model from this thing? So what does that mean? Let me go through the steps. First, you measure the response of that neuron in the monkey brain to a few hundred natural images, like this, random images of the kinds of things that at least people see. Then you look at the selectivity, the response of that neuron to these images. And so this is a response of one neuron shown in black, and there's a bunch of different categories of objects in these images. And this is a neuron that's face selective, right? So all of these stimuli here are faces, and you can see it responds strongly to all of those images. This has been known since the 1980s that those neurons were in there in the brain, okay? And so then, now we have the response of that neuron. Now we measure the responses of the artificial neural network. What does that mean? It means we feed the same images in the front end, let them funnel through the network, producing activations, collect activations from a late stage of the network, and then we try to fit a predictive model based on those ANN activations. So what does that mean? That means we come up with a weight, separate weight for each of the units in that layer, a few hundred, maybe a few thousand units in the layer, depending on the network. For each unit, we fit a weight such that when we multiply the activation of that unit times its weight and sum them all up across here for a given image input, that equals the response of the neuron. Okay? And so we're just using this as a kind of equation, essentially. Put an image in, get some activations, run it through here, and have it fit these responses. So you do that on all of the images but one, and then you freeze these weights, and then you feed in the held-out image, and you ask how well you can predict the response of the held-out image. Okay, when you do that, you find that here are the predictions in red for each of those images, and they pretty much exactly match the observed responses. Each of these predictions was based on a set of weights trained without that image, so it's not overfitting, right? It's cross-validated. So what does all that mean? That means we can take these trained artificial neural networks and use them as a prediction device with a kind of weighted combination to predict how a neuron in the brain will respond. Okay, it's evidence for some kind of similarity between convolutional neural networks and brains, but what kind of similarity is a wide open question, much debated, but to the extent that we think it's evidence for some kind of similarity, it's a beautiful echo of an idea that David Marr, visionary vision scientist, said way back in 1982. What Marr said was that the nature of the computations that underlie perception depends more on the computational problems that have to be solved than on the particular hardware in which those solutions are implemented. And so he didn't know that someday artificial neural networks would be able to do these human-like tasks and would be able to fit responses in the brain, but essentially what he's saying is it's the structure of the problem that's going to constrain the solution, and here we see some kind of evidence for that. These artificial neural networks have nothing to do with brains. They weren't modeled to look like brains, and yet they arrive at some kind of similar set of functions in here. Okay, but really what I want to do is now use these things to test the selectivity of those brain regions. That was all background. Okay, so how about these face, place, and body regions? Are they really selective? The trouble is, the claim of selectivity is essentially a claim over the infinite set of other possible images we could present, that none of them will produce as strong a response as faces do, or places or bodies in that part of the brain. And needless to say, I haven't tested an infinite set of other stimuli. I've tested, a few dozen, maybe a few hundred, and it's hard to know which ones to test after that. But now, and so we don't know what to test, how to refute this, right? So, but now what we can do, this is work with my former postdoc Ratan Murty and Jim DiCarlo, now we can use these predictive models, do the same game I just explained, try to build a predictive model of the response of these whole regions, and then use it. So let me show you how that goes. First question is, can we build a predictive model of the response of a whole brain region? And the answer is, oh, yes, we can, to an astonishing degree. So on the x-axis is the predicted response of the fusiform face area to each stimulus. Each dot is a stimulus, okay? And on the y-axis is the observed response that will be observed when we actually scan people looking at that image, okay? Again, as before, these predictive models, the weights of the predictive models were trained excluding that image, right? And so what this shows us is that we can predict very precisely with a correlation of over 0.9 how each of these regions will respond to a novel image that we haven't scanned it to before. And that's pretty amazing. But is it just a stunt? Well, we can use it. We can use it to test the selectivity in a more turbocharged way than has ever been possible before. And so what we did was we ran the entire machine learning image database of 3 million images through these predictive models over a weekend. And what I was actually hoping is that this is like a really powerful way to disprove your hypothesis. So I was really hoping that we would find some images that were predicted to produce strong responses in the face area model that weren't faces, Because then it was like, okay, now I know what to test. Let's scan people and see if this is really a violation of the hypothesis. But Ratan and I hand-checked the top 100,000 images for each, and we could not find any of the top images for the face model that weren't obviously faces, for the place area model that weren't obviously places, or the body area model that weren't obviously bodies. There's one kind of exception. One of the top images for the body selective region was this. It's an axolotl. I'm giving it a pass, close enough, but there's some judgment call in there. The other thing you can do is you can use generative models to produce images designed to maximally activate these regions. And we do that, when we do that based on our data, we get this for the face area, that for the place area, and that for the body area. They're not exactly, and this is like, 4 years ago. They'd probably be much better now. Pretty good. So the point is that this is a kind of, fancy AI way to use AI tools to more powerfully try to falsify your hypotheses, and so far they haven't been falsified. Okay. All that is still looking at just the responses of these regions, but really what we want to know is, are they causally involved in behavior? Remember that a highly selective region that responds very strongly to faces doesn't mean you need that region for face recognition. It kind of makes you wonder, makes you think you might, but doesn't show that you do. So that's why a few years ago I was pretty excited when a colleague told me that this gentleman was awaiting neurosurgery in Japan and had electrodes all over the bottom of his brain. Here are the electrodes on the bottom of his brain. And he had kindly agreed to allow us to collect response. This is, awaiting neurosurgery for epilepsy. And he had kindly agreed to allow us to collect neural responses from these electrodes while he looked at our images. And so just for comparison, here's the bottom of my brain showing you my face areas in red, my place areas in green, and some color-preferring regions in purple. So you can see that those electrodes are going right over what looks like it would be some of the good stuff in this guy. So we sent some images, and they sent us some data. And just for some examples, here are 2 electrodes right there in a region that looks a whole lot like my fusiform face area. And each plot shows you time on the x-axis, this is like 2 seconds, and response now measured from actual electrodes on the surface of the brain to faces in red and to lots of different stimulus categories in all these other colors. And so if you were unimpressed with that bar chart I showed you at the beginning, it's like faces, other things, like not that impressive. That's because functional MRI is a very blurry method. When we have the actual real deal, these regions are spectacularly selective for their preferred categories. Okay, but we haven't gotten to causality yet. That's just background. How do we test causality? Well, the neurosurgeons very occasionally decide that they need to electrically stimulate parts of the brain in order to test the causal role of that region in behavior. And naturally, we don't get to choose, we scientists. We just get to be happy when we get the data that's informative. Of course, these decisions are made based on clinical reasons. But these neurosurgeons were considering neurosurgery in the vicinity, and they wanted to know what happened when they stimulated here. So I'm going to show you a video of this guy being stimulated in a face-selective electrode. He's speaking Japanese, but the sound probably.
One more time.1秒ぐらい始まって1秒ぐらいがなんか横になんか目口って感じでちょっと一瞬見えたんですけど、これ何かなと思ってたら気が付いたらもうこの箱にしか見えてないっていうか、なんですかよまたさっきみたいに横にその目と口って感じでちょっとアイスなんですか ごめんなさい、もう一回。
One more, please. One more time.
やっぱり顔に見えました。目、口らへんって感じ。
Okay, these kind of data are rare and precious. We don't usually get to have a neurosurgeon stimulate in exactly the region that we're interested in, but they're very powerful. They show us that that region is not only selectively responsive to faces, but causally involved in face perception, and not causally involved in the perception of other things, other than to stick a face on top of them. Okay. All right. And so there's little specks of data like this for the other regions, but that's some of the best data we have. Okay. Next question. It's hard to look at a picture like this, at least for me, and not wonder what other specialized regions are in there, and in particular, for what mental functions. Okay. So you may have noticed that many of these regions are for various social functions, which is not surprising. We are social primates. We care deeply about other primates, and here are all the social ones. But it's also true that a lot of what we do is not social at all. So if I show you an image like this, you quickly see, okay, there's a fork, there's a glass, there's a table, there's orange juice, there's a placemat, but you also see more than just a list of objects. You see the glass is supported by the table, that it contains orange juice, that it could be picked up, but might spill, and then would stain the placemat, right? And probably a lot of that occurred to you just when you just looked at it within a first second or so. And so the point is that perception is a lot more than collecting a list of categories of objects that are present and their location in the visual field. It also involves determining physical relationships between things, action affordances, what you can do here, and possible future states. Okay, and so the broader picture is this is a part of what you might call everyday intuitive physical reasoning. And the idea is that we do this like all the time. We do it frequently, rapidly, effortlessly, and apparently automatically. In fact, you cannot take a single action on the world without some kind of mental model of the physics of the world you're acting on. If I want to pick up this laptop, I need to first have an idea of where to grasp it. Where are the stable grasp points? What does it weigh? What is the friction? If I want to walk across the room, I realize that I better not keep going over here, or there might be trouble. All of those kinds of action decisions are based on a prior model of the physics of the world. And so this whole area was first studied in rich detail by developmental psychologists like René Baillargeon and Liz Spelke, who pointed out that these things are developed very early. They're essential for understanding what we're looking at, planning action, and predicting what will happen next. They are also, by the way, at the root of our quintessentially human ability to devise novel tools, which itself is, at the root of our species' signature triumphs in engineering, architecture, technology, etc. So I think these are pretty important. And the question is whether there's specialized brain machinery for intuitive physical reasoning. So we've been looking at that question for a few years. And this is all work with my colleague Josh Tenenbaum, starting off with Jason Fisher. And Jason did some functional imaging experiments a while back. And what he found was that there's a systematic set of regions, shown here in 4 subjects, in the parietal lobe and frontal lobe, that are systematically engaged in a bunch of different situations. First, we showed people these rotating towers, little videos, and we asked them to decide if this tower falls, will there be more blocks on the red side or the green side of the platform? That's a basic intuitive physics task. You got to figure out where's it going to fall, where the block's going to land. Versus we had subjects do a different task on the same stimuli. We just asked them to say whether there were more yellow or blue blocks, okay? And so that doesn't require much in the way of physics, and we balanced it for difficulty. And in that contrast, we get these regions in pretty much every subject. So we thought, okay, that's nice, but who knows? Let's try another experiment. So then we made videos of moving dots. And some were like the classic Hyder and Simmel movies some of you may have seen, where the dots are moving around in a social fashion. They're chasing and flirting and interacting. And you just see these moving dots, and you just know this is a social interaction between these 2 agents. Versus a physics condition where they're bouncing off each other and off the walls, and it's obvious that they're inanimate and just bouncing physically. In that contrast of looking at physical dot motion versus social dot motion, we get the same regions. So that's nice. And then all of this led to this broader hypothesis, which really comes from Josh, who wants to say that these brain regions, which we're calling the physics network, actually I keep telling Josh we should call it the hypothesized physics network, and that causes him to whine and say, do we really need to still say hypothesized? And I say, yes, Josh, we do, but it didn't fit in the slide, so I took it out. Anyway, still hypothesis, that these regions implement our understanding of the physical world, which contains some kind of generative model that can run forward simulations of what will happen next. Kind of like the physics engines that are built into video games. So when you play a video game, it needs to know if you whack something in this direction, it's going to ricochet off of here and fall off of there. It needs to have a model of physics built in to generate those visual stimuli. And so Josh's idea is we have one of those in our heads. And I must say, when he first started telling me this, I thought he was completely out of his mind. But after enough repetitions, I said, Josh, it's an empirical question. Let's just test it. And here's our first two things. Okay, so if these things are really part of the brain's physics engine, then they should contain all kinds of information relevant to physics. They should contain information about the mass of objects, about whether objects are in contact, whether the configuration of objects is stable, and they should be able to make predictions about what's going to happen next. And I think what I'm going to do, since I think I mistimed this talk, I'm going to skip over the details and just tell you, for those of you who know how this works, that using pattern analysis methods, and I'm happy to chat with anybody offline about this since I'm skipping over it. Using pattern analysis methods, we've shown that you can decode whether an object is heavy or light from the pattern of response in those regions. Those regions contain that information. They contain it abstractly. The pattern for a heavy versus a light thing is the same, whether it's an object falling into a bowl of water or landing on a pillow that tells you its mass. They also contain information about whether a scene is unstable like this or stable like that. And they also, and this information, importantly, is not present all over the brain. We don't see it in the ventral visual pathway where you have face, place, and body regions. We don't see it in primary visual cortex where you have basic visual information. We see it up there in those candidate physics regions. We also show that, I don't know how long I'm supposed to, I'm just going to cut this for now and we can come back to it if there's, you can ask me if there's time in the questions. Yeah, okay, I'm gonna cut this and we can come back to it. And I'll just say that we've also shown that you can decode from those same regions whether objects are in contact, and we can also decode a future prediction of what's gonna happen next. And so where we are is some preliminary evidence. We certainly haven't nailed it yet, but for a set of regions up in the parietal and frontal lobes that seem to be implicated in intuitive physical reasoning that you might think of as the brain's physics engine. Next, we need to do lots of other things, determine the scope of engagement of these regions, test computational models of what these regions are doing, test their causal role in behavior, and all of that is ongoing. But so we get to add some blobs, maybe. Okay, but what I want to do is get to these other questions about how does all this organization arise in development? I find it just impossible to look at pictures like this and not wonder, how do you build that? This is a basic structure that's present in something like this form in pretty much every normal person. How does it get built? Okay. And so I'll focus first on the fusiform face area, and there's about 20 years of papers in which people have argued that the fusiform face area takes many, many years to develop and requires visual experience. Okay, but that's mostly based on scanning people at age like 7 and up, and showing that there's some subtle changes in the fusiform face area between age 7 and adulthood. And the problem with that, okay, they've gotten down to 4. Problem with that is that's still too late. That's too old, right? Infants are already good at face recognition actually they can recognize faces within minutes of birth to some degree, but they're even better within a few months to a year. So looking at what happens after age 4 is too late. Really what we need to do is scan infants, and not only scan infants, but scan them while they're awake. And this is nearly impossible. But luckily I have intrepid colleagues who figured out how to do this, and that would be Rebecca Sachs and her then grad students Ben Dean and Hillary Richardson, and Rebecca's son Arthur. And collectively, over many years, the four of them figured out how to get sensible data out of infants. It was quite a struggle. I get no credit for this whatsoever. I just watched from a distance and thought they were nuts, but that it was cool. And then eventually, they got data like this. So this is data from a 6-month-old infant showing differential responses when that infant is looking at images of scenes versus faces. And these patterns are roughly similar to where you get those activations in adults. So that's pretty cool, but it doesn't enable us to tell really how selective these regions are or to nail the hypothesis of what these regions are doing. So that's when the really amazing Heather Kosakowski joined the scene. She was a grad student with me and Rebecca, and the first thing she did is decide that you can't just stick infants in normal adult head coils, you get lousy data. So she designed her own infant functional MRI coil. It's this cool device where you put the infant in and then bring in these plastic sides, slide in all comfy and snug, because head motion is a big problem with infants. And even more amazing, in the course of one PhD, Heather scanned 83 awake infants, and it's just impossible to express what a feat that is. Every one is kind of a train wreck, you know, and she just kept going. And the whole essence of this is about 10% of the data you collect is actually usable, and so you just have to prevail and prevail to get that little bit of data where there's little enough motion that you can see anything. But it paid off. Heather found face, place, and body areas in 6-month-old infants. And here in held out data is the selectivity of the face area. Purple bar is response to faces. The other bars are responses to bodies, objects, and scenes. Place area is weirdly below our control condition, which is a whole saga, but the response is higher to scenes than the other conditions, and the body area produces a higher response in held out data to bodies than other conditions. So all that says is, and actually we've pushed this down to 3 months. So these regions are present by 3 months of age. So no, the FFA does not take years to develop. What about, does it require experience? Okay. Many people have tried to go at this in various ways. It's obviously a hard question. But Ratan decided to go at this by asking whether you can ever see a fusiform face area that's selective for faces in people who have never seen. So he decided to scan congenitally blind people while they felt 3D printed stimuli. Okay, so here's our high-tech device looking into the scanner bore. There's a disc of cardboard with some 3D printed plastic things, and a postdoc stands outside the scanner bore, rotating the cardboard every few seconds so the subject can feel the things as they go by. Here are the stimuli, 3D printed faces, scenes, hands, and chairs. And here is what we see in the ventral pathway in sighted subjects looking at those stimuli, faces versus objects. That's the usual fusiform face area. Here's what we see in the congenitally blind people, feeling faces versus feeling other things. It's weaker. It's present in a little over half of subjects, not all of them, but it's eerily in the same place. And when you look in held out data, the selectivity is very similar for the blind people feeling faces as the sighted people seeing them. Okay, so apparently you do not need to see faces to develop a selective region for faces in your ventral visual pathway. That doesn't tell us how this organization arises, but it gives us some importantly constraining data. It arises really, the face area arises really early and does not require face experience. Okay? Okay, last question. Why is this a good way to design a brain? I used to end every talk with this, and I just thought that was just kind of fluff, because actually I wondered, but I didn't think it was anything we could ever answer. And this is one of the reasons that I've gotten excited about artificial neural networks, is I think that they can give us a really powerful clues into these questions. So Katarina Dobbs was a postdoc in my lab doing the work I'm about to describe. She's now has her own lab at the University of Gießen in Germany. And so what we did was we took a convolutional neural network, this one with more layers than the one I showed you before, details don't matter, and we trained it to identify 500 classes of faces, face identities, like Joe versus Bob versus Mary, whatever, and 500 classes of objects. And we mixed all the training stimuli in the training data. So this network was just trained to classify these images into these categories, okay? And so we didn't tell the network, hey network, these are faces, these are objects. We just mixed them all together, okay? And so this network knows nothing about the significance of faces to humans. It's just given a bunch of visual patterns and it has to assign them to different output categories. And so I thought this network would do badly because we were giving it these kind of two problems, and how was it going to grapple with these two very different kinds of problems at once? And I thought that would be sort of loosely consistent with the fact that the brain uses different machinery. Well, that prediction was totally wrong. The network did great on both face identification and object classification. And at first I was like, damn it, I really liked that hypothesis, but I guess it's wrong. But then, as one does when one disproves one's hypothesis, one thinks a little bit more. So we thought a little bit more, and we thought, well, let's look inside that network and see how it's doing that. And so we performed lesion experiments on the network, which you can do just like studying patients with lesions, except you get to control where the lesions go. It's awesome. And so we took a late layer of the network, and we found a set of units in that late layer such that when you lesion them, performance on the face discrimination task gets really bad, but leaves the object classification task intact. And we found another set of units, when you lesion them, you totally screw up the object's ability to classify objects, but you leave intact its ability to identify faces. So this is a double dissociation, beloved of neuropsychologists for many years. And here we have it in a network, and it's showing essentially spontaneous segregation of the network into two separate systems, one for processing faces and one for processing objects. And we did not tell it to do that. It discovered it on its own, and it has no idea what these patterns mean. It doesn't know, oh, social primates care about faces, need to engage with that. It doesn't know that at all. They're just patterns that it's classifying. And so just based on the statistics of its input, it has spontaneously discovered this organization that we see in the brain, kind of latent in its structure. Okay, what about other categories? So I'm leaving out some of the details because some of this is a little bit technical, but this network was trained supervised, where we gave it labels for each image. That's fine, but it's kind of, I don't know, 2010s or something like that. And further, more seriously, there aren't large enough data sets of object classes that are labeled so that we can do this with all the different object classes. But luckily, the machine learning people have invented this cool thing called self-supervised training, where you do not need labels on the images, and it uses very generic rules to try to train up the network just based on the statistics of the input. Okay, so next we decided, let's use these self-supervised networks just trained on natural images, and let's see what structure arises. Okay, and let's ask whether more generally do human-like representations arise in artificial neural networks trained self-supervised on natural images. Okay, so first what we did was to look more systematically at some brain data, from some functional MRI data from humans. And for this, we used this magnificent public data set that Kendrick Kay put out a few years ago called the Natural Scenes data set. And usually, I don't like to mess with other people's data because I figure mine are better. But Kendrick Kay's data are really awesome. He scanned these subjects on 7 Tesla machines, and each subject looked at 10,000 images, some of them many times. It's a major playground. I recommend checking it out. So we decided to take Kendrick Kay's beautiful data and use some data-driven methods to just kind of look at those data and do some math that I'll leave out for now. That math just asks, what are the dominant response profiles in this whole pattern of data across the whole ventral visual pathway over these 10,000 images? And when we do that in humans, we get a bunch of components out that the math is telling us these are the dominant components. And then we look at the images that produce the highest response in each component. And what do we see? We see faces, scenes, bodies, and words, all of which had been characterized before. And people in my lab are like, why are you so excited? You already knew that. I'm excited because these things emerged from data-driven methods in a data set that I did not collect to make myself happy, right? They're just there. They're just dominant properties of the response of the ventral visual pathway. The food thing is funny. We and others published claims from these data that there's a food selective response in the ventral visual pathway. I now don't think it's exactly food. We don't know what it is. It's very food related. I'm just going to call it food for now, but just know that it's actually not exactly food. Okay, so this is still looking at human brains. You may note these white spaces. These are the most consistent things that emerged from the data-driven analysis. But there's some other stuff. There's often other stuff. Sometimes you stare at it, you can't make head or tail of it. I don't know what these other things are. But in any case, we can now apply the exact same methods to artificial neural networks trained self-supervised on natural images and ask, what is the dominant set of response profiles in artificial neural networks? Okay? Okay, and what do we find? We find a bunch of dominant components, just like we did for humans, and when we inspect them, I'm sorry, these images got really small. The top ones are faces, places, bodies, food, and text. This should be very surprising. I find this absolutely astonishing, right? These networks were not even given labels for these images. They just did this kind of very rudimentary, kind of generic training on natural images. And after training, they developed a representational structure that is shockingly similar to the human ventral visual pathway. These networks have no domain-specific priors built into them. They have an architecture. You've got to have some, thing to train. They have natural images, which, yes, were based on where humans decide to park a camera and take a photo, so all of that probably matters. But they are not given any categories. They know nothing about the meaning or significance of these categories to humans. And to me, that says we need to really question whether we need to build in domain-specific priors to account for development of these regions in humans. It's kind of unparsimonious to say we need that when the networks do the same thing and they didn't have any of that. Okay, I don't know if I went over, but I will end there by saying, why is this a good way to design a brain? I don't know exactly, but I think the fact that kind of recognizably similar structures emerge spontaneously in artificial neural networks that have minimalist priors and certainly no domain-specific priors should make us should make us think that one, there's probably deep computational reasons why these things arise, and two, you do not necessarily need to build in domain-specific priors to get them. And I will stop there and take questions and go back to some of the stuff I skipped if you like. So thank you.
Is working, but
just
if you
talk
have
loud.
any questions, please, raise your hand.
Okay. Was that a question? Okay. You? Yeah.
Super interesting talk. Thanks so much. Your experiments on infants and blind suggested that maybe experience not visual experience maybe isn't super important for facial plasticity in the brain, whereas your experiment on the artificial.
They're just totally contradictory. You're absolutely right.
What's your take on that?
I don't know what to make of it, but you're absolutely right that these things are kind of going in opposite directions, right? Like in humans, you don't really need the experience. Now, there's other experience, there's tactile experience, right? I don't think that's enough to account for it in the blind necessarily, because blind people don't really recognize each other by feeling faces. When we first started this work, I kind of Googled it, and I found all these web pages set up by blind people who say things like, how come sighted people are always asking if we want to feel their faces? That's disgusting. So it's like not a thing, right? But that's not to say that they don't have some tactile experience. I'm sure they feel their own faces. They probably felt their parents' faces when they were infants. So there is some face input, just not visual face input. And so, there's a little bit of wiggle room there. I don't know how to reconcile this. The ANN stuff doesn't tell us how it works in humans. It just says you should be aware of the possibility that you don't necessarily need to build in domain-specific priors. It's just kind of an in-principle argument that you might not need it. And so kind of computational parsimony might suggest that we should work on, hypotheses that don't build it in. But yeah, it's very hard to put all the fact that it's present early, I think, is cool, but doesn't nail it. Because, even 3-month-old infants have spent a lot of time looking at faces. So that's hard to tell. Using which for language?
Just for the people in the back, he said, do you think the question holds for language as well?
Which question? Early development or ANNs? Okay, so there are ways to look at that. I think they're ongoing now in various labs all over the place. I'm not doing that stuff myself. I'm trying to remember. Okay, if you look of course you can do all this stuff with transformers and large language models and all of that, right? If you take one of the classic large language models, like GPT-2, which I think was when they were still trained on only text, then you can't really sensibly ask whether it does other domains of stuff because it's only experienced text. But the more modern ones that have been trained on the entire internet, the images and the sounds and the text and the whole shebang, then you could say, okay, that's really broad training data. What is the internal organization of those things, right? And so there's some very recent papers. I saw one that came out just like a week ago, I haven't read it yet, arguing that those broadly trained large language models segregate aspects of linguistic knowledge from arithmetic knowledge. So I don't know, it's interesting. The point is it's now a kind of tractable kind of domain of computation to look at. I will say when we first found the spontaneous segregation of faces and objects, I was so thrilled. I mean, it was just so astonishing to me. My first reaction was, once we figure out how to train a network on the task of life, it should recapitulate, of human life, it should recapitulate the entire organization of the human cortex. And I think that may be true ultimately, but that's obviously a little hard to do.
Thank you. I enjoyed your presentation very much, and I have a pair of questions. One first, starting from your initial slides, where you mentioned that that activity, neuronal activity, cognitive function are more driven, task-driven, functionality-driven, whereas the underlying dialectic plays a little role. I'd like clarification of that, because
I didn't quite hear you. Just say it again. What is the claim that you want more on? Just say.
Yeah, sorry, you heard nothing I said? Or?
No, I heard most of it. I just.
Oh, okay. Yeah, so at some point, the dialectic has to play a role. If you don't have the underlying media, you cannot generate those impulses. So I'd like a little bit of clarification on that statement. The second one has to do with the.
Yeah, I can't do two. I can't hold one question in mind.
Sorry, we'll do one
okay,
at a time.
Okay, but I don't understand the first question yet. Just can you ask it a different way?
Yes. So the first question is, can you clarify what the first statement that you made about the hardware? I suppose cognitive function being hardware independent.
The David Marr quote. Yeah. Okay. I mean, I didn't make a strong claim about it. I just said it's notable that we find similarities between convolutional neural networks, which are very different in hardware, from brains, and that are optimized with totally different processes, backprop versus evolution and natural selection plus learning, right? So the basic hardware and the training rules are wildly different. Nonetheless, both are kinds of task optimization processes. And apparently, in at least some cases, when you start with these very different physical systems with very different training optimization methods, if they're optimized for the same task or a similar task, they come up with similar solutions. That was the claim.
Okay, understood perfectly. The second question having to do with the face and object recognition being independent of any training. Does that mean that a condition like prosopagnosia is tied to very specific genetic biomarkers, and should we be looking for those rather than looking for neuropsychological?
Well, it's not either or. We should look for all of those things.
All of them.
Right, and so, prosopagnosia, there's acquired prosopagnosia where you have damage to that region and lose your face recognition ability. There's also developmental prosopagnosia where there's no known brain damage, and these are just people who've just never been able to recognize faces at any point in their life. And there's very interesting work on all of those fronts, especially on the developmental prosopagnosics. Most of them do have a fusiform face area. It's a little bit smaller, it's apparently not sufficient to have a little face selective region to be able to recognize faces. Whether that's because the circuit can detect faces but not discriminate them, and that's enough for an MRI signal, whether it's because the output connections from that region are disrupted. There's a little bit of evidence for each of those things. There's also work on genetic bases of developmental prosopagnosia. It's not very clear last I checked. Face recognition ability is heritable. That is, it's more similar between two identical twins than two fraternal twins. That's suggestive, but also extremely ambiguous, because the relevant genes could be, for example, genes for extroversion, right? So if you're more extroverted, you look at faces more. So maybe it's a genetic effect that's really based on an experiential effect. So there's little shreds of evidence, but none of them nail those questions.
Excellent. Thank you very much.
Yeah.
Yeah, I'm just curious because you said you like psychology a lot. So I'm curious, like, what about this interests you in psychology? Like, how do they connect together for you?
Yeah, it's a great question. Well, my whole background is in cognitive psychology. My first year in graduate school, 1981, the first brain imaging study was published on the cover of Science, and it was this big blurry mess that showed you like a blob for V1 when people were looking at patterns, and the blob was not there when they had their eyes closed. And I just thought, I have to do that. But it took me 10 years, literally 10 years of beating on the doors before anybody let me do a brain imaging experiment. The technology was controlled by radiologists, who, I'm sorry, there's probably radiologists in earshot, but honestly, they do not know what an experiment is. And so they published horrible study after horrible study after horrible study in Science and Nature. And me and my psychologist friends were tearing our hair out on the sidelines saying, we actually have theories and questions and hypotheses to test. Please, 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 please. So at the most general level, the sense in which I think this is relevant to psychology is just the broader question that's been around in cognitive science for, depending on how you count it, like at least a century, of domain specificity of mind. Like, do we have completely generic minds that are just kind of equipotential for any old thing? Or are minds composites of distinct mechanisms that do different things? And I think on that question, I think these data are really powerful. They're not the only kind of data that are powerful. Of course, we had, centuries of neuropsychology that already gave some pretty powerful clues. But to be able to go in and systematically test each of these things and say, okay, what exactly does this region do? I think it's another way to discover natural kinds of the mind and brain, and that's really what I'm in it for. I don't really care if the thing is here or there or what it's shaped like. I care, how is it carving up the space of cognition?
Thank you so much.
Somebody had a question back here in the, yeah.
So hello, thank you very much for your talk. Is this working?
Yeah.
Okay. I did have a little more questions on the self-supervised model because I feel like we skipped on it a little bit.
Did.
So can you tell me about like what the task, what the architecture was, and also what's the whole correlation thing?
Okay, so we've done it a bunch of ways. And there's several papers that have been, that published this before we got our stuff out. That's fine. Everybody's doing similar stuff here. So I would refer you to Margalit and Yammans et al., 2024. It's a beautiful paper. That one builds in other constraints like spatial smoothness across a layer, which is a whole different thing, trying to get the actual spatial structure of activations in the brain. There's a really nice paper by Prince and Conkle and others. So there's a bunch of papers out there. What we did was we did it a bunch of different ways. We trained them, in one case, on ImageNet. These are trained on an image set called EcoSet, which is designed to be a little bit more like human experience. It's still just a big machine learning data set of millions of images, but they're supposed to be, it's not like 30% dogs or whatever ImageNet is. ImageNet has really uneven distributions of different categories. Okay, so we trained, I believe this is ResNet 160, I forget. It's one of the later ResNets, trained on EcoSet, and it's trained self-supervised, which basically means you take each image and you do a bunch of augmentations of that image. So you turn it to grayscale, you crop it in different ways, you rotate it, you flip the color axes, you do all of these kind of affine kind of transformations, you stretch it, change its size, you do all these kind of transformations you can do bottom up on an image, and you just train the network to treat all those augmentations as the same. So the training just says, feed these different images through and make sure they produce similar representations at a late layer. So that's all that goes into the training. It's a very generic training rule. It knows nothing about what the categories are. It doesn't know that this dog image that you started with and augmented in all these ways is the same category as some other dog image that you augmented in various ways. It just somehow discovers that. It's pretty astonishing.
Okay, and but in the end, the output was a set of categories that arose from your data set.
Okay, so find me afterwards and I'll tell you in more detail, but just briefly for now, there's lots of ways to look for these categories. We wanted a data-driven way to characterize the dominant response properties of either the ventral pathway in the brain or the artificial neural network layer. And so we use something called Bayesian non-negative matrix factorization. Basically, you have a bunch of voxels by stimuli, or units in the network by stimuli, with an activation in each cell, okay? And so it's just basically matrix factorization. So, you know, PCA is one version of that. It makes assumptions we didn't want to make. You could use ICA. Those work kind of sort of, but we think NMF is a more appropriate way to do this. And when we apply that matrix factorization, it basically distills that matrix into a small set of components that account for much of the data in the matrix. And these are those components.
Thank you. I will find you after this.
Yeah, okay. Yeah, there's somebody back there. Yeah. Recognize faces, whether that's because the circuit can detect faces but not discriminate them, and that's enough for an MRI signal, whether it's because the output connections from that region are disrupted. There's a little bit of evidence for each of those things. There's also work on genetic bases of developmental prosopagnosia. It's not very clear last I checked. Face recognition ability is heritable. That is, it's more similar between two identical twins than two fraternal twins. That's suggestive, but also extremely ambiguous, because the relevant genes could be, for example, genes for extroversion, right? So if you're more extroverted, you look at faces more. So maybe it's a genetic effect that's really based on an experiential effect. So there's little shreds of evidence, but none of them nail those questions.
Excellent. Thank you very much.
Yeah.
Yeah, I'm just curious because you said you like psychology a lot. So I'm curious, like, what about this interests you in psychology? Like, how did they connect together for you?
Yeah, it's a great question. Well, my whole background is in cognitive psychology. My first year in graduate school, 1981, the first brain imaging study was published on the cover of Science, and it was this big blurry mess that showed you like a blob for V1 when people were looking at patterns, and the blob was not there when they had their eyes closed. And I just thought, I have to do that. But it took me 10 years, literally 10 years of beating on the doors before anybody let me do a brain imaging experiment. The technology was controlled by radiologists, who, I'm sorry, there's probably radiologists in earshot, but honestly, they do not know what an experiment is. And so they published horrible study after horrible study after horrible study in Science and Nature. And me and my psychologist friends were tearing our hair out on the sidelines saying, we actually have theories and questions and hypotheses to test. Please, 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 please. So at the most general level, the sense in which I think this is relevant to psychology is just the broader question that's been around in cognitive science for, depending on how you count it, like at least a century, of domain specificity of mind. Like, do we have completely generic minds that are just kind of equipotential for any old thing? Or are minds composites of distinct mechanisms that do different things? And I think on that question, I think these data are really powerful. You know, they're not the only kind of data that are powerful. Of course, we had, you know, centuries of neuropsychology that already gave some pretty powerful clues. But to be able to go in and systematically test each of these things and say, okay, what exactly does this region do? I think it's another way to discover natural kinds of the mind and brain, and that's really what I'm in it for. I don't really care if the thing is here or there or what it's shaped like. I care how is it carving up the space of cognition.
Thank you so much.
There's somebody had a question back here in the, yeah.
So hello, thank you very much for your talk. Is this working?
Yeah.
Okay. I did have a little more questions on the self-supervised model because I feel like we skipped on it a little bit.
Did.
So can you tell me about like what the task, what the architecture was, and also what's the whole correlation thing?
Okay, so we've done it a bunch of ways, and there's several papers that have been, that published this before we got our stuff out. That's fine, everybody's doing similar stuff here. So I would refer you to Margalit and Yammans et al., 2024, it's a beautiful paper. That one builds in other constraints like spatial smoothness across a layer, which is a whole different thing, trying to get the actual spatial structure of activations in the brain. There's a really nice paper by Prince and Conkle and others. So there's a bunch of papers out there. What we did was we did it a bunch of different ways. We trained them, in one case, on ImageNet. These are trained on an image set called EcoSet, which is designed to be a little bit more like human experience. It's still just a big machine learning data set of millions of images, but they're supposed to be, it's not like 30% dogs or whatever ImageNet is. ImageNet is has really uneven distributions of different categories. Okay, so we trained, I believe this is ResNet 160, I forget. It's one of the later ResNets, trained on EcoSet, and it's trained self-supervised, which basically means you take each image and you do a bunch of augmentations of that image. So you turn it to grayscale, you crop it in different ways, you rotate it, you flip the color axes, you do all of these kind of affine kind of transformation, you stretch it, change its size. You do all these kind of transformations you can do bottom up on an image, and you just train the network to treat all those augmentations as the same. So the training just says, feed these different images through and make sure they produce similar representations at a late layer. So that's all that goes into the training. It's a very generic training rule. It knows nothing about what the categories are. It doesn't know that this dog image that you started with and augmented in all these ways is the same category as some other dog image that you augmented in various ways. It just somehow discovers that. It's pretty astonishing.
Okay, but in the end, the output was a set of categories that arose from your data set.
Okay, so you can find me afterwards and I'll tell you in more detail, but just briefly for now, there's lots of ways to look for these categories. We wanted a data-driven way to characterize the dominant response properties of either the ventral pathway in the brain or the artificial neural network layer. And so we use something called Bayesian non-negative matrix factorization. Basically, you have a bunch of voxels by stimuli, or units in the network by stimuli, with an activation in each cell, okay? And so it's just basically matrix factorization. So, PCA is one version of that. It makes assumptions we didn't want to make. You could use ICA. Those work kind of, sort of, but we think NMF is a more appropriate way to do this. And when we apply that matrix factorization, it basically distills that matrix into a small set of components that account for much of the data in the matrix. And these are those components.
Thank you. I will find you after this.
Yeah, okay. Yeah, there's somebody back there. Yeah.
Yeah, thank you so much for your talk. I have a question regarding the artificial neural network that you mentioned when you classify different neurons.
Can you speak up a little bit?
Okay.
Or just hold it closer? I heard the artificial neural networks, yes?
Yeah. So my question is, since it's unsupervised, which means, it's supervised, which means like humans will give the labels to different, like based on their neural profile. I wonder, like, is it important for you to make sure that the classification is exhaustive, so that
Sorry, that the classification is what?
it's exhaustive?
I don't know what you mean by exhaustive.
Like it covers as like comprehensive as possible, so that you like don't miss out a potential classifications that you didn't consider? I don't know whether.
Okay, I'm just not hearing well, but I think what you're asking is, what is our coverage of the space of images? Is that essentially what you're asking? No?
Yeah, kind of.
I mean, look, let me just answer that, and then you can chime in with whatever I don't get. I mean, look, it's not like, this artificial neural network that generated these components is the only possible way to do this or the end of the story. Of course, it's trained on some set of images that were taken by humans and that people uploaded to the web and some, computer science people collected in a big database, right? And so that has all those biases in there, right? So there's lots of dogs and faces and stuff like that. But the EcoSet version of it is an effort to at least somewhat even out those categories. Actually, what they did was they took word frequencies, like how often in human text do people use the word dog and chair and tree and such, and they use those words frequencies to determine the frequency of those images in the water. That we haven't gotten very far. But one can ask now all kinds of interesting questions about what needs to be in the training set to get this, right? And so, for example, we thought, this is a whole sidebar, but we thought that the food component was related to color. And so we thought, okay, here it is emerging in a very human-like way, and they're both very colorful. Does it emerge as a distinct component because the food tends to be colorful in this image set? So we just converted the entire training set to grayscale images, trained a new network, self-supervised, and we get the same food component. So no, doesn't need color. So that's just an example of how we can explore what it is about the training data that's essential for these components to emerge. And there's no presumption that these will always be the components. They just popped out of our first couple of efforts, and very similar stuff have happened in other people's work, but it's wide open for further exploration.
Yeah, makes sense. Thank you.
One last question.
Yeah, okay. I'll just talk out loud. Were these responses able to categorize some multiple? Were they able to respond to multiple categories? Like, would faces ever show up in bodies, or would these?
Yeah. Okay. These components that emerge, both from the brain and from the networks, sorry, I skipped over all of this. I just showed you some top image. A component, you take this matrix, you factorize it. Each component is a response profile over the stimuli and a distribution over voxels or units, whichever you analyzed, okay, that enable you to reconstruct the original matrix you started with, okay? So if you look at those response profiles, for example, the face one, they're really face selective, right? We used 1,000 images for this analysis because those were the ones every subject in the database saw. And the top, I'm just guessing, but certainly the top 50 are all faces. There is some gradient after that. And so there will be eventually some interleaved thing where there will be, some, I don't know, a dog or, some other stuff and then another face. So it's not perfect. It's not like a step function that all the faces are ones and everything else is 0. There's a gradient like that for each of these categories. Nonetheless, when you look at the selectivity, like you look at the top images or any kind of the other thing we do is we look at how correlated are they with human ratings of the salience of that category, of that category. So we show people all the images and we just say, rate how salient a face is in this image. And if it's a picture of some park with nobody in it, the number is like 0. And if it's like a close-up of face, it's like a 10, right? So we get all those ratings. And those ratings are strongly correlated with these components.
Thank you so much again, Dr. Kanwisher, for this fascinating talk. Honestly, it's such an honor to have you. We're truly grateful that you took the time out of your day to come all the way from Boston just for this conference. So really, I still can't believe that this is happening, and it doesn't feel real, but really, thank you so much. This
My
has
pleasure.
been incredible. So let's give her another round of applause.